Okay, thanks very much to the organisers for letting me speak today. I'm a population geneticist, which basically means I go out and gather lots of data. So I'm really blown away by all the amazing lab studies and clinical studies that I've been seeing today. And saying that, please bear with me while I try and persuade you that the data that I gather is actually important for vector control. So I work out at Silwood Park in Austin Burt's lab, and I'm part of the target malaria team that Alikos introduced the project earlier. So we haven't, slight problem with my slides there. We haven't heard a huge amount about vectors today, but basically the ones that we're most interested in targeting in Target Malaria Project are the Anopheles gambiae complex. And these are responsible for most of the malaria in sub-Saharan Africa. So the three main vectors are gambiae, that should say, Kalutzii and arabiensis. And then there are some local vectors as well, Meris and Melas, which are found along the east and west coasts of Africa, respectively. Bwambi, which is a very local vector just found in saltwater springs in Uganda. And then there's also a couple of non-vector species. And these all make up the Anopheles gambiae species complex of very closely related species. So these two at the top, Gambiae and Kalutzii, are very, very closely related. And in fact, they were only designated as species in 2013. Before that, they were known as chromosomal or molecular forms of Anopheles gambiae. So a lot of the time when you hear people talk about gambiae, they are actually talking about those two species and not distinguishing between them. So in 2013, Kalutzii was given its name, and previously that was the M form. So we first got our reference genome for the Anopheles gambiae in 2002. It's a pretty good reference genome. It's been assembled to chromosome level, saying that it is unfortunately a chimera of the M&S forms, so the gambiae and the Kalutzii, because it was a lab strain that was sequenced. Following that, there's been some technical challenges for doing next-generation resequencing of whole genomes from individual mosquitoes, because you can imagine these are very, very small organisms and you don't get a lot of DNA out of them. But now we've got to a stage where we can routinely sequence the genomes of individual mosquitoes without using whole genome amplification. So the first genome studies that appeared around 2010 mainly used pooled mosquito samples and usually from lab colonies. But now we have the first large-scale study, which will soon be published, using whole genome data from individual wild-caught mosquitoes. And that is actually published. It'll be published soon in Nature, but following on from one of the other speakers earlier, I'm pleased to say that this has already been put onto bioRxiv in an early non-peer-reviewed form. So it's already out there for anybody to see that work. So of course, before we could do all this population genomics, we were already interested in the population genetics of these mosquitoes. And there were lots of studies using mitochondrial DNA, microsatellites, etc. But of course, they are limited because there are only a small number of loci that you could look at in that way. And in just a moment, I'm going to show you some of the first results from the 1,000 Genomes Project, which will show why this is quite important, because you really need to know which loci to select for what you're looking at. There were some pretty reliable paradigms that came from the pre-genomics work. For instance, there's very high genetic diversity in these species, which implies a very large population size. There are very few barriers to gene flow, surprisingly, considering these have a fairly short dispersal. But you don't see any barriers to gene flow across continental Africa except at the Rift Valley. And we also know that there's some gene flow between these very closely related species, Anopheles gambiae and Kalutzii, and possibly with arabiensis as well. So why do we care about this? So for these new methods that we're developing for vector control, not just in target malaria, but there are various other labs, including other labs here at Imperial and elsewhere, that are developing these genetic technologies, including gene drive, which are really, really promising methods for vector control. And because these are genetic methods, we need to know how this gene will move through a wild population. So we need to know something about what's going on out there in the wild. We need to know if there are going to be any barriers to gene movement spatially, which is kind of obvious, but less obvious perhaps is temporally, because in a lot of places where there's transmission of malaria, there are these very severe dry seasons where mosquitoes seem to disappear. So we need to know whether releasing our mosquitoes before a dry season, whether they will actually persist and the genes will carry on through the dry seasons. We also need to know whether we have cryptic breeding units in our populations, because most of these mosquitoes are morphologically identical, and the only way you can tell whether you have any structure in your populations is by looking at the genetics. It's also important for us to know something about the population size of our target population. This may sound like an easy thing to go out and survey mosquitoes, but it's actually really hard to estimate population sizes just by surveying. We are attempting mark-release recapture experiments, but they're incredibly labour-intensive. So if we can develop methods, genetic-based methods, where we can just sample a small sample of mosquitoes and look at the genomes and make an accurate estimate of population size, that would be a really great thing. And we need to know that population size so that we know how many of our transgenic mosquitoes we need to release to have an effect on the wild population. And also so that we can effectively monitor size change after a control intervention. So I'm going to present a little bit of data from the Anopheles gambiae 1000 Genomes project. And this is the project that's about to be published. The first phase of it actually analysed 765 Anopheles gambiae and Anopheles coluzi from across Africa, from 10 populations across Africa. Target Malaria collaborates with the Anopheles gambiae 1000 Genomes project. We contribute samples, and we've also had some involvement in the analysis of the data. So this is a really nice analysis that the project did to show how different parts of the genome show different predominant signals of evolutionary history. And along the top here is a representation of the chromosomes, so chromosome 2, 3, and X. And along the bottom here are just some examples of neighbour joining trees that have been made from the SNP, the polymorphism data in sliding windows along the genome, looking at 100,000 bases in each window. And there's some, depending on where you look in the genome, you get these sort of more predominant signals coming through. For instance, in some regions, you have inverted regions, which are big chunks of chromosomes that have flipped around, and the orientation that they're in is the strongest signal that you get if you create a phylogenetic tree in that part of the genome. In other parts of the genome, the main signal you get will be geography or demography. And in other parts of the genome, the main signal you get is the differentiation between the coluzi and the gambiae samples. And then added to that, you'll get some areas, for instance, near the centromere on 2L, where you get a very, very strong signal, which comes from the insecticide resistance allele, the KDR allele. And what the 1000 Genomes guys have done here is paint these windows to show you which is the most predominant signal in each part of the chromosome. So that shows how important it is to select your SNPs and your loci properly when you're doing some sort of analysis. So this is some work that they did just using those SNPs, which are geographically informative. So they got rid of anything that was in inversions or in these sort of regions of speciation. And they did an admixture analysis of this for each population, which basically clusters. There's a clustering analysis, and the different colours here show the amount of, give an indication of whether these are separate populations, whether they can be distinguished as separate populations. And along the bottom here, this shows the actual sample sites with pie charts that are colour-coded according to these clusters. And the sort of the take-home message here is that these three populations here mostly cannot be distinguished from each other. And this is actually a distance of around 3,000 kilometres. So there seems to be very little barrier to gene flow across this region. There is some differentiation in the Cameroonian samples, which is associated with the sort of ecology of where they were collected. So some of them were collected up in the sort of more drier sort of, I think you call it Sahel sort of region, and others were down in a more moist area. And then you get this differentiation as well when you take samples from the other side of the Congo Basin. So we already knew about a barrier to gene flow in the Rift Valley, but these samples have also showed that there is a barrier to gene flow in the Congo Basin. However, you can also see there's still big gaps, huge gaps, especially in the middle of the continent there. So the 1,000 Genomes Project is going to go on to do a lot more sampling, a lot more geographical sampling and temporal samplings, and of different species as well. So there's a lot more to be done. So I mentioned that we need to know about population size. So very sort of crudely, high genetic diversity equals a large population. And the 1,000 Genomes Project found incredibly high diversity in these samples, with 50 million variants over 141 million base pairs of accessible genome, which equates to about one SNP every 2.2 bases. And so we know that they're big populations, but really what we want to know is something a lot more accurate than that for our project. We want to get a good idea of what local population sizes are like. And we're going to be using a lot of different patterns of genetic diversity, so not just doing a simple SNP analysis. I won't go into all these in detail, but there are various different measures of genetic diversity that you can use, including association of SNPs along the chromosome. And we have people in our lab working on developing new methods to more accurately estimate population size from this sort of data. We can also use the population genetics data to look at changes in population size. So this is actually some simulations that I did of population crashes of different size, the sort of size of crash that you would expect to see after a vector control, and at different times in the past. So this is generations in the past. And by looking at these different population parameters, this is a linkage parameter, this is a diversity parameter, and these are other different diversity parameters. And by looking at these different measures, you can, and taking samples through time, so before, during, and after your control interventions, you should be able to get a good idea of the population size change. And as I said, the advantage of doing something like this is you could literally take 12 mosquitoes from your area where you're doing the information, analyse those genomes and get these figures. So it's a lot less work than going out and attempting to do these really involved, labour-intensive surveying techniques. Another thing that's relevant to vector control, which Alekos already mentioned earlier, was the issue of resistance arising to your genetic construct. So we need to know how much standing genetic variation there is likely to be at the target site in our wild populations, and that will help our modelers to determine how quickly resistance may arise. There's also an issue of resistance arising actually due to the mode of action of the construct. And we can try and minimise that effect as well by using areas of the genome that have very, very low polymorphisms that are highly constrained. And that would mean that even if the construct actually does damage that bit of DNA, it'll probably have such a strong fitness cost that it wouldn't cause resistance to arise. So this is some work that's ongoing at the moment. And this is just a sort of an example of a sort of alignment that I can, or sort of chart that I can make, where I've got the genomic positions, the bases that are present at those positions, and then I have various different variation data from different data sets. So from an alignment of 21 Anopheles genomes, 9 Anopheles genomes, the 1,000 Genome Project data, or from the G3 colony. And I can make this sort of chart and pass it on to our genetic engineers, and they can choose target sites from this. So another thing that we want to know about is whether we're going to have gene flow between species. So we know from past genetic studies that there is sometimes gene flow between Anopheles gambiae and Kaluzi. You do find quite a lot of hybrids in the wild, especially in some areas in West Africa, but those hybrids are not just sort of dead ends. The genes do actually continue to flow between the species. And insecticide resistance genes have been a really good way of seeing the sort of dynamics of that movement between the species. And there's a really nice study that you can have a look at, which is a time series of samples taken in Mali, where right in the middle of the time series, you'll see this big chunk of chromosome 2 that moves from Gambiae into Kaluzi, taking the insecticide resistance gene with it. We also want to know whether there's gene flow with other species. So a recent study by Michael Fontaine showed that there has been historical gene flow between Gambiae and arabiensis, but we don't have any evidence at the moment that that is still ongoing. For instance, we don't see that jump of insecticide resistance genes between those two species. However, arabiensis has been quite understudied with regard to population genomics at the moment, and there is a plan to add that to the next round of the 1,000 Genomes project. So what we still need to know is we still need to know about fine-scale population structure and dispersal. So we're using our collaboration with the guys from the 1,000 Genomes project to do a lot more sequencing on fine scale in our potential release sites. And we've taken on a new postdoc who's going to do some modelling of changes in allele frequencies to try and get to grips with fine-scale population structure and dispersal. We also want to know about these temporal barriers to gene flow and also small-scale barriers to gene flow. And again, we're going to be using genomic data to do these things. We need to know at our own study sites the local population sizes and the diversity of the target site, and gene flow between species. We need to know all these at the actual sites where we're going to hopefully implement the controls. And we also want to know about these constraints on target sites, so that we can get an idea of fitness costs if the construct causes any mutations to the target site. And again, that can be fed to the modelers to give us an idea of resistance arising. So that's it. I've just got to say thank you to our funders, the Bill and Melinda Gates Foundation, Open Philanthropy Project, and the Wellcome Trust, which funds the 1,000 Genomes project. Target Malaria is a big team, so I can't name everybody. So I'll just mention the boss, Austin Burt, who unfortunately couldn't be here today. But thank you.